you about the preprocessing. Here, we also calculate other quality control metrics, such as the total counts, the percentage of mitochondrial genes per cells, or the doublet detections. So the next steps correspond to normalization. We are trying to make data comparable despite the difference on the sequencing depth. So we are trying to reduce the biological variation we found on single-cell RNA data in order to understand our data, or our data set, sorry. The next step corresponds to dimensionality reduction. Here, techniques like PCA, TSNE, and UMAP help you simplify this high-dimensional data into lower dimensional representations while preserving this cell relationship we found on this high-dimensional space. So this is crucial because we are always dealing with thousands of genes across thousands of cells. The next step corresponds to clustering. Here, we're trying to group cells based on the gene expression profiles. And here, we identify the cell types or state present in our sample. And we also have the ability to understand the variability within our samples. And finally, we found the differential expression analysis. Here, we are trying to identify marker genes that can define the cell population or response to the conditions present in your data. So these standardized pipelines allow single-cell RNA-seq data to be reliable, analyzed, compared, ensuring reproducibility and meaningful results. However, mastering a programming languages like Python or R can be really challenging. So this is where Single-Cell Explorers comes in, offering a user-friendly platform in order to reduce the complexity of this pipeline without any coding skill. So Single-Cell Explorer is built on 4 main technologies. First, we found Docker that help us to reproduce and isolate the environment of each one of these components. That means that researchers or users can run the same analysis without any compatibility issues. We try Single-Cell Explorer on macOS, Windows, and Linux, and we obtain the same results. The second technology is the backend. We developed this using a Python library called FastAPI that allow us to create application programming interfaces. In the case of Single-Cell Explorer, we developed more than 30 endpoints in order to integrate both Scampi and Seurat in the same data pipeline. So with this, we leverage the best of both programming languages. The next technology is the front end. Here we used Node.js, a framework from JavaScript, in order to create the graphical user interface that you want to see later. And finally, we found the SLOM that corresponds to the research management. So here we can efficiently manage computational resources and scale analysis jobs dynamically across multiple nodes. So all these components create a scalable and user-friendly platform that reduces the gap between a computational method and biological research. The first step on Single-Cell Explorer corresponds to upload your data. Here you can choose between several formats, both for Python and for R, and you have to define the analysis, the name of your analysis. You also have to define the species of interest. Currently, we have available for human, mouse, and zebrafish. You also have to choose the gene ID. We currently have available gene symbols and ensemble ID, and you can also use your email to get the result, and you can run it later if you need it. We also count with a tutorial that is present in each step of this pipeline, allowing the user to understand more in depth each one of the parameters present in Single-Cell Explorer. We also count with 3 examples data sets corresponding to human PBMC samples, mouse cortex, hippocampus, subventricular zone cells, and zebrafish cranial neural crest-derived cells. So once you upload your data, you will see a quality control metrics corresponding to your raw data. It's also important to mention that in every step of the pipeline, you can export your data set, both for Python or for R. So now in the preprocessing, we are going to use the quality control metrics previously calculated in order to apply filters based on the number of gene byte counts, the total counts, and the percentage of mitochondrial genes. For that, we have parameters like the mean of genes, the mean cells, that help us out to eliminate low-quality genes and cells. We also found the mitochondrial threshold when we set a maximum of percentage of mitochondrial gene per cells. This is because we already know that higher values of percentage mitochondrial genes are indicator of dead cells. So we're trying to exclude that from our analysis. And also, we include for the doublet detection, this is where 2 cells are encapsulated and sequenced together. And we use for this the scRNA library on Python. that help us to eliminate this technical issue. So after you apprise run this, or you filter based on these parameters, you will obtain your pre-processed data, and you can always rerun the analysis until you obtained the biological question you are interested. Next, we found embedding. Here, we have to define the number of high variable genes. This is really important in single-cell RNA because the high variable genes help us to separate the latent space in order to understand how many cell types or sites are present in our samples. Also, we have two methods to understand or to select these high variable genes. We have CRAT method and the cell ranger method. So after you apply these parameters, you will obtain the quality control metrics previously calculated, but now over a PCA. We also, additionally, we also include an elbow plot to understand how many principal components we are going to use for the non-dimensional reduction analysis, such as UMAP. And we also include a class tree that is a library from R that helps us to understand how many, the resolution, or in other words, how many cell types can be present in your samples. So with these parameters that we calculated before, we can now perform the UMAP. For that, we are going to use the information about the component, the principal component calculated before, and the latent resolution. And we also found another parameters corresponding to the number of neighborhoods that help us to adjust the latent space created by UMAP. So higher values give you a broader view of your manifolds, and while lower values give you a more local space data. So the results from this is going to be your latent cluster, or the cell types present in your samples, and all the quality control metrics over UMAP. The next step corresponds to the differential expression analysis. Here, users can define the number of top genes present in each cell types and statistically determine using or with Coxon sum test or t-test. Additionally, users can define or can manually input the genes present in their datasets in order to understand the biological question of interest. So the result of this corresponds obviously to the log fold change, or a dot plot from the log fold change of each cell type identified. And this is correspond to the log fold change. We also are able to calculate the mean expression values. The next step corresponds to visualization. Since we are already calculated two-dimensional reduction analysis, we can use UMAP or PCA in order to visualize up to 6 genes per visualization. So this is important because we are not always interested on the high variable genes. Maybe you are interested on other genes, and you can see and visualize here on this step of the pipeline. And finally, once you analyze one sample from single-cell RNA data, it's really common to try to integrate this with other samples or other studies. However, batch correction is an issue in single-cell RNA data that prevents us to separate ourselves based on biological difference, and cells are finally separated by technical issues. So for this, scExplorer addressed this problem with 4 methods for batch correction corresponding to Compact, Scanorama, BBKNN, and Harmony. And we create this in order to, the user can run all of this and see which one is more suitable for your type of data. So as a summary, scExplorer can read several inputs for single-cell RNA data, allow you to pre-process your data consisting on quality metrics, doublet detection, and data normalization. You can perform dimensionality reductions such as PCA and UMAP, differential expressions analysis such as mean and log fold change, and also with the nerves, I forget to tell you that all the plots here are dynamically presented because we are using the library Plotly, so you can gain real-time insight about your data. And also, you can export your data set both on Python or R in each step of the pipeline. Here you have a QR code. We have the server running like about 2 months and already helped some researchers to understand the data. I want to acknowledge the fundings that allow me to be here presenting this work, and the NetBio Lab and Immuno-Oncology Lab for their support on this work. Thank you very much. Thank you.
multiple parameters in the different steps.
Yes.
Have you considered exporting? Exporting those parameters so that it's easier than to present that in an article?
I'm so sorry, I'm not able to.
Exporting the parameters that you chose during the process, the whole process, so that then it's easier for
Yes,
methods.
Yes, I understand. I think that one thing that missed the application is that I don't show you here is that finally you have a results page when you can download all your data, but doesn't count about the parameters. I think that is something that we can integrate later to improve the analysis.
And it would also be useful to import parameters so that you don't need to.
Yes, I don't think about that. Yes, it's a really good question. Thank you.
Well, I think you already answered. So you can download the pre-step file, so I can use it for analysis by myself on Python.
Exactly.
Well, this is computationally very heavy. So my question would be, how many transcriptomes or how many samples can we analyze using this tool?
Thank you for your question. The samples dataset that I show you here are corresponding to like 2,000 and 5,000 cells within 2,000 genes, 20,000 genes. But I tried my application with a huge dataset with more than 100,000 cells, and we don't have any problem running with the server. The only thing is that because we have too many data and the plots are dynamically presented, we have to present you only static plots because the web server goes down if you try to analyze that amount of data.
The analysis will be deleted after.
Also, it's a really good question. Thank you. Data is stored on 2 weeks, then it's deleted.
You mentioned some methods to reduce the batch effects. I don't know if have you ever seen or noticed a difference between using several pipeline and using these methods.
I understand. Actually, one of the methods I present you here for the batch correction corresponding to Harmony is from R. The other, Combat, I think it's also present in both language programming. In the case of Scanorama and BBKNN, this is all, just present in Python. I don't know if that answers your question. Thank you. Any question?